Thank you very much for coming. We're here to talk about civil society call for a global moratorium on the use of gene drives. The global moratorium was signed by over 160 civil society organizations representing environmental groups, labor groups, small-scale farmers, indigenous people, scientists, and social groups. This is a call for a moratorium on genetically engineered gene drives, and it's a call that's coming from six continents and 127 countries. The groups that have signed this moratorium include Friends of the Earth International, International Union of Food Workers, La Via Campesina, which is here representing today as well, International Federation of Organic Agriculture Movements, the International Indigenous Peoples Organization, TEBTEBA, Network of Scientists for Social and Environmental Responsibility, and the Unions de Científicos Comprometidos con la Sociedad de México, ETC Group, and Third World Network, just to name a few. So why does civil society care about this call for a global moratorium? The use of gene drives contradicts the moral purpose that we have in conservation organizations. We want to protect nature and ecosystem integrity, and we want to support food sovereignty around the world. Gene drives threaten both of these. Gene drives are transboundary, and they are transgenerational. All people need to be included in strong and diverse dialogues about how to govern this technology, given that it will impact everyone. So who should decide that we permanently alter the world around us? Given the dire situation that our environment is in, we need to act with precaution and in the interest of people. So the statement of this moratorium urges governments to adopt a global moratorium on the genetically engineered gene drive, which poses serious and potential irreversible threats to biodiversity, peace, and food security. Civil society is asking national and international governments to protect biodiversity, indigenous people, and local communities' rights from the impacts of controversial new biotechnologies, such as the gene drive. I'd like to read the gene drive moratorium as supported by civil society groups. In the view of the significant ecological, cultural, and societal threats posed by genetically engineered gene drives, including threats to biodiversity, national sovereignty, peace, and food security, we, the undersigned, call upon governments and the 13th Conference of the Parties to the Convention on Biological Diversity, in accordance with the precautionary principle, to put in place a moratorium on 1, any further technical development and experimental application of gene drives, and 2, environmental release of genetically engineered gene drives. Thank you.
Good afternoon, everyone. I'm Ricarda Steinbrecher, representing the Federation of German Scientists. I'm a biologist, I'm a molecular geneticist, and I'm going to present why, from a scientific perspective, this moratorium call is urgent, and it is in line with the precautionary principle. This idea of wanting to eliminate species that are causing problems, whether these are pests, whether weeds, invasive species, carriers of diseases. This idea has been around for some 50 years, with the hope one would find a way of genetically being able to manipulate the pattern of inheritance. Basically, Mendel's 50-50 is not good enough in order to get rid of these species. The idea was one would need something that can be driven through, that would be passed on to every generation, to all offspring. Now, CRISPR-Cas has become a tool around genome editing, and it is the perfect tool also to now be used within the setting of gene drives. What it does is you will cut a piece of DNA at a very precise place, and if that place you choose is a gene that, for example, then will be disrupted and now there will be no female offspring, or there will be other disadvantages, then that would be the target gene. With the CRISPR-Cas, you cut it and you make a string, a DNA segment that you place in there, which actually is the instructions again for these molecular scissors. that now will be passed on to the next generation, who will build the scissors, cut the genes, insert it, and this way you spread the information. You basically are breaking the rules of genetics, genetic inheritance in that. Now, what is the problem here? For one thing is, it is still a model. We should not forget that. It was only last year that for the first time it was proven to actually work in laboratories. But it is very new and we have no idea how to assess it. Now, if we are going to release it into the environment, or if it gets there accidentally, which question should we be asking? Because we shouldn't forget that all species out there are part of networks, of ecosystems, ecosystem functions and services. Even mosquitoes are, even mice are, they are part of food chains. So invasive species in a farmer's field or in the ecosystem, they have a home. An alien invasive species comes from somewhere, where it is a plant or an insect or whatever organism it is, where it is there on its own and it's on its own merits, and it is part of the system. So if now I send a gene drive after these organisms, this will ripple through all the ecosystems back home, where it will, of course, also have a major effect. What it can do, if you take out one species, it will shift the ecosystem balance. If you alter it, it will shift the balance. How can we predict what it does? We can't. We do not have the knowledge to do so yet. So therefore, there needs to be a way to actually ask the questions, collect the questions, understand what we know, what we don't know, how to actually do risk assessment here. And in that meantime, it would be most unadvisable, would be irresponsible to start releasing. And in that way, we come to the point of saying the precautionary principle has to be enacted. Until we have all the relevant ways in place to do risk assessment, until we know how to answer the questions, we need a moratorium, saying that it is too risky to have these organisms released into the environment. Because, for example, with mosquitoes, you could have the problem, you take them out, something else replaces them. That could be even more tricky, even more dangerous. You could have it with other species. You remove them, something else fills in it. We have had that with other pest management systems. You eradicate one, the next ones come, and they often are worse. So we could actually exacerbate problems rather than giving a solution. So that needs a lot of research, and in the meantime, a moratorium is the right answer.
Thank you. My name is Jim Thomas. I am Program Director with the Etc. Group, an international organization that tracks emerging technologies and their impacts on society and on biodiversity. And I'm just going to give a little status update on where we are on gene drives. So the idea of a gene drive, while it's been around theoretically for many years, it's really only since the beginning of 2015, less than 2 years, that gene drives have existed. The last time that the COP met in Korea, there was no such thing as a working gene drive. It simply did not exist. And as a result, we're looking at probably one of the fastest, maybe the fastest emerging technological controversy I've certainly ever seen, probably this century. In 2015, at the beginning of 2015, a team at the University of San Diego, led by Ethan Beer, created a gene drive in fruit flies that turned fruit flies yellow. And it was done within a laboratory. Unfortunately, they never asked for any biosafety approval from their university. And this was in a location, California, where fruit flies are in fact endemic. And had one of those gene drive fruit flies escaped out of that laboratory, it's theoretically possible it could have spread across the North American continent and beyond. It didn't, thankfully. Since then, we now have gene drives have been put into a number of species. They've been put into yeast, they've been put into nematodes, they've been put into mosquitoes, and there's work at the present at North Carolina State to put them into mice. This is in less than 2 years. And at the same time, we're seeing tremendous amounts of money being funneled into developing gene drive systems. The Gates Foundation, through the project, the Target Malaria Project, has put $75 million in. The Tata Foundation, associated with the large Indian conglomerates Tata Industries, has put around $70 million in. And overall, the most visible funder that's emerging is the US military. The Defense Advanced Research Projects Agency, DARPA, has a couple of projects that are about developing gene drives, or rather developing ways to deal with rogue gene drives in the environment, to deal with gene drives going wrong or gene drives escaped. It would not be appropriate for DARPA, the defense agency, to be developing offensive gene drives, because that would be in contravention of at least two international conventions, the Bioweapons Convention and the 1977 NMOD Environmental Modification Convention. It's still worrying, however, that this is being driven very much from military interests, and certainly there are potential military uses here. And very quickly, we've seen efforts to try and understand the governance implications of gene drives. The National Academy of Sciences in the US mounted very quickly a large study that came out in June this year, a 200-page study, looking at the governance questions around gene drives, and some of the clear implications are there are very large implications for biodiversity. These gene drives should not be being released into the environment at this time, or deployed certainly. The question of gene drives was actually raised at the ARTEG on synthetic biology. It also came up at the World Conservation Congress in Hawaii in September. And there, in a motion on synthetic biology, the overwhelming number of both governments and NGOs called for what amounted to a de facto moratorium on any IUCN support for gene drives. And that came at a time when 30 leading conservation and environmental leaders, including Dr. Jane Goodall, Dr. David Suzuki, Dr. Vandana Shiva, who just won the Midori Prize here in Mexico, all called for no place for gene drives in conservation. There is a very high level of alarm about gene drives. The gene drive community, which is now growing, there are more labs that are experimenting with this technique, are beginning to say, maybe we can develop ways of making these local so that they don't spread globally. Can we make gene drives we can recall, gene drives that will stop working? All of this at this point is very speculative. The concern, of course, is that with so much money flowing into this field, with so much effort to develop gene drives beginning to proliferate, and with the underlying technology of CRISPR-Cas9 remarkably simple and easy to access, it won't be too long before a gene drive gets out by mistake. So this then becomes an important transboundary issue. And I'm going to pass over to Lim-Li Ching from Third World Network, who can talk about how this fits into this convention. Thank you.
Thank you very much. My name is Li Ching. I'm with the Third World Network. I just want to make 3 points. The first is that genetically engineered gene drives are designed to spread and persist. They will therefore have no respect for national borders. Whether deliberate or accidental releases will undoubtedly have transboundary effects. Secondly, there are as yet no international governance structures or regulations that can adequately and effectively deal with the situation. While under the Convention on Biological Diversity, we have the Cartagena Protocol on Biosafety, which is focused on transboundary movements of LMOs. The rationale of this law, all its provisions are really about stopping the spread of transgenes. So there is nothing at the moment at the international level that is equipped to deal with the unrestricted flow of a living modified organism without respect for borders and even entry over multiple borders. It is because of this international governance gap, as well as the serious ecological and societal effects that gene drives release could introduce, that is why we are calling for a moratorium. And the CBD, the Convention on Biological Diversity, is the international legally binding treaty addressing biodiversity. As we all know, its objectives are for the conservation of biological diversity, its sustainable use, and the fair and equitable sharing of benefits arising from the utilization of genetic resources. Its provisions oblige parties, of which it has almost universal membership, 196 parties, to address any threats and adverse impacts of biological diversity. Therefore, this call for release, for moratorium on the release, development, and research on genetically engineered gene drives is really the inappropriate response, and parties can take action now at this meeting here in Cancun, in Mexico, under the discussion that is currently occurring on the agenda item of synthetic biology, as gene drives are quite clearly an application of synthetic biology. Thank you very much.
So if there are questions, we'll take those. There are representatives of some of the signatory organizations in the audience as well. I think the format is just an open forum for questions. Any questions? Got a microphone in the back. Clarifications?
I would like to add one point, which Jim just brought up, that of course we cannot predict whether it works or to which extent it will work. And one of the problems is, of course, CRISPR-Cas being used as a genome editing tool, and very often exactly the information that the genetic information that inscribes, that codes for CRISPR, being introduced into that experiment, there is a danger that accidentally you can create gene drives. And that is actually amongst the scientific community quite a worry. So that is something which we need to look at additionally. The other aspect, of course, is with CRISPR-Cas as gene drive design, it could also be that after a time, it will actually not work because, in a way, the organisms will build resistance to it, and it will then sort of come back. And what that means for diseases once they have started resisting, when they have started to build up sort of an antidote, they often change traits in that as well, or vigorousness. And so that is also aspects we need to take into account when looking at risk assessments.
Okay, thank you. My name is Maureen Bostock, and I'm here representing La Via Campesina this afternoon. And I just want to say that farmers from my country in Canada, as well as in every country that La Via Campesina is present, are very concerned about this new technology, as we have been concerned about genetic engineering being so unregulated for the duration of its existence. In particular, also, we're very concerned about the issue of transboundary, and in particular, the rights of Indigenous people around the world to free and informed prior consent. So I wondered if someone on the panelists could talk a little bit about that issue and how the delegates at this convention could address that issue. Thank you.
I mean, I think one of the key issues with this whole idea of gene drives, really, as you've pointed out, are the transboundary effects. If a gene drive organism is released in one country, it will spread. It will have no respect for national borders, and therefore, countries, let alone indigenous peoples and local communities, will have no say. If this is allowed to go ahead, they will have no say as to, or to have their consent sought, whether or not they agree with such a release or what the implications are, particularly the socio-economic implications on certain species and for peoples. And this is why it's such a major issue, and why we are saying, look, we do not have the procedures in place yet to deal with this. This is unprecedented. If we talk about GMOs as we know them, the Cartagena Protocol on biosafety, its very rationale is really to, have a procedure that really intends to stop the spread of transgene. It's only for the intended users, yeah? But with this now we have potentially uncontrolled spread, and therefore things like free, prior, and informed consent will go out the window, really.
If I may give a very concrete example. So one of the reasons why the topic of gene drives became an issue in Hawaii, where the World Congress, Conservation Congress met, was because there was a proposal, and in fact work going on at the University of Hawaii, to release gene-drive mosquitoes in order to attack mosquitoes that are spreading avian malaria that affects a particular very important bird in Hawaii. This idea that you would release mosquitoes for conservation purposes to remove an invasive species was being promoted by a North American biotech NGO. But it was very much opposed by local indigenous peoples in Hawaii. There were indigenous groups who were saying that this is our territory, these are our birds, this is we have our own ways of managing the forest, and the way in which we should address the loss of species should be from our own knowledge. In fact, the groups in Hawaii have actually been sending postcards to the convention here saying we don't want gene drives in Hawaii. We want to have control and decision-making over our own conservation decisions. So that goes to this question about indigenous control over territory.
Sí, buenas tardes. Soy Marlene Castro de la organización Periodistas de a Pie. Mi pregunta es, bueno, han dicho que este asunto de las herramientas como parte de la biología sintética es algo que no se puede predecir, pero sí podríamos establecer escenarios, o sea, ¿cuál sería el peor escenario y cuál sería, en el mejor de los casos, el menor escenario?
I think we can both share it. It's a good question with very little backing in terms of very little data to actually give you an answer. Because exactly what I was saying is that what is the worst case scenario is something we cannot even imagine in the moment. What it could, for example, relate to is that you have ecosystem shifts and therefore collapses, that therefore, for example, you could have a shift of insects which could bring in like more disease carriers. You could have, if you remove mice, you could like alter the food chain drastically. When this gets out to the natural population, mice is a prime food source for birds. It's like part of the food chain. Just imagine they are not there. So it would have like global knock-on effects for all ecosystems here. We shouldn't forget we are at a time when biodiversity is really at a very vulnerable stage. We are losing it rapidly. Ecosystems are becoming more and more vulnerable because they are less and less biodiverse, and it just takes a very little to knock them, and we depend on them. So the scenarios could be like if it's being used in agriculture, that it just backfires, and that which we have had so often with technological approaches, that it will do a moment, it gives a breathing space, and then it crashes down on us. But in order to give you specific answers, we would actually need to look at the specific gene drive system that one would think of, or that is intended, and then study where are these organisms all present, where are they part of the food web, where are they part of ecosystem services, how are they interacting with others. If we take them out there or they are altered, what would be the knock-on effects? It would require quite a big research in order to be able to answer the questions.
If I may add just one piece to it, I think in order, before we talk about what would be an okay scenario, in addition to needing to understand the questions, the risks and the impacts on the environment, it's also really important that we have a very robust public dialogue. This is not just about the impact on plants and animals and insects. This is about the impact on people's livelihoods, their ability to have control over their own food system, to depend on and live with natural resources. And we need to be able to have this conversation amongst many people. And remember that part of the evaluation is socioeconomic impacts, and that's a key part of what delegates will be discussing here at the negotiations. That socioeconomic impacts and cultural impacts are a key part of biodiversity. So before we talk about what are possible benefits, is there a scenario where this could be safe? We need to understand way more about the science, but we also need to be able to think about, is there ever going to be a regulation, assessment, oversight that could adequately or even come close to what's necessary to control a technology like this that knows no boundaries, that's meant to spread through populations? Is this something that we'll ever be able to do responsibly? And we're not yet at that question, and we need to look at a diverse set of impacts, not just about the environment.
Thank you. Just the question, as I heard it, was what is the best case scenario? You're going to hear the best case scenario from the organizations and individuals who want to move ahead with this, who will give rosy views of this being a predictable technology. So Island Conservation, who want to release gene-drive mice by 2020, and who will make out that this is a predictable, stable technology. They'll give you the best case scenario. The problem is you're dealing with life on many levels. You're dealing with it at the genetic level, where we have levels of unpredictability, and you're dealing with it at the ecological level, which is also an unpredictable level. So the best case scenario shouldn't be the basis on which we make decisions. Precaution requests that we also look at some of the worst case scenarios, and that decisions are made collectively, because this affects us all.
Other questions?
Yeah. Well, obviously this topic is related to the precaution principle, and as far as I know, only the European Union has incorporated this principle to their legislation. So I want to know if we can think of that maybe in the long run we can incorporate this precaution principle into the international treatments and the national laws.
The precautionary approach actually has been recognized since the 1992 Rio Declaration, and it is actually in described in the Convention on Biological Diversity and the Cartagena Protocol on Biosafety. In fact, Cartagena Protocol on Biosafety operationalizes the precautionary principle. It doesn't name it as such, but it's in there. So very much so, the CBD operates under the guidance of the precautionary principle.
Yeah, but it's only a guidance, right? So, because it's only a principle, so that's a big question if we could enforce it.
Well, I mean, in this case, in fact, there are existing decisions on synthetic biology in which all the countries to the CBD have said that they will apply the precautionary approach on considerations of synthetic biology, especially releases to the environment. So in this case, there's an agreement between all the member state or the parties of the CBD to apply the precautionary approach.